All right. I cannot stress enough the vocabulary for this particular chapter. If it feels like I've been speaking in tongues in the last two chapters, it's only going to feel a little bit worse in this one. So before you even go through this lecture, I would strongly recommend you take a few minutes, go look at the vocab list that I gave you, and see if you can write down the definition of those major words. Before you start this lecture. If you can have a rough understanding of that, maybe go watch a few of the Amoeba Sisters videos because they can animate everything. I'm going to do my best, as always, to define and explain everything to you. But the more that you come in prepared for it, the better this is going to go. Okay? All right. But let's get started. So for this chapter objective, there is a lot here, but again, most of this is going to be vocabulary related, as well as just understanding the major components of genetics. You could study your whole career on genetics and never understand everything. So when we're saying we're doing this in one chapter, 2 lectures, it is because we are going very surface level on this. However, we're going to talk about a lot of it from the interesting perspectives and the things that we can physically see and experience as humans. But keep in mind that this is a huge topic. So one chapter is not enough to really get into genetics. Okay? So, who was Gregor Mendel? Gregor Mendel was an Austrian monk, and he worked in the garden in the 1860s. He was actually someone who was very into statistics. He was a mathematician, and he worked in the garden as part of his chores and something that he enjoyed at the monastery that he went in. At the time that he was doing his work, he just started noticing patterns in the garden. And he really chose pea plants, and that's not the only thing that he studied. That's not the only plant or organism that he had made notes of, but his pea plants are best known and understood because the variations were so clear, and he was able to do a lot of tests and a lot of replications of his tests with the pea plants. Peas are a fantastic test model because they actually can grow from seed to harvesting in 60 days. And there's a ton of variation in here, but we generally see 2 types of characteristics for each of the traits. And so he was able to actually do a ton of research on this. At the time that Gregor Mendel was working, most people knew that both sexes contributed to a new individual, but the understanding of meiosis was not there. Our full understanding of reproduction was not there, but we did know that like a mom and a dad came together and made a baby, right? If I bred a black horse and a white horse, I'm going to get a horse that has some of those characteristics. There was some understanding of that. But we didn't understand genetics. Obviously, Gregor Mendel did that. We did not know what chromosomes were. We did not understand meiosis. So it's really cool that he was doing a lot of this way before our full understanding was. And at this point in time, a lot of science was passed on through lecture circuits. And some of the things that I've read are that like he just wasn't great on the lecture circuit. He was a little bit awkward. He stumbled over his words. He didn't make eye contact very well. And he wasn't really good with people, which maybe he was a little neurospicy, right? Like maybe that was a thing. We also know that Gregor Mendel had sent his work to Darwin. So Darwin was actively working on his hypothesis about descent with natural selection, and we're going to talk about that descent with modification in the next unit. But Darwin never read Mendel's work. It was found in a locked drawer after Darwin's death, and it would have actually answered a lot of his questions that he had that would have supported his understanding of his idea of descent with modification and would have sped up our understanding of evolution. In addition, Mendel's work was almost lost. After his death, his work was locked away, and it was almost destroyed when the monastery was torn down. But it was found and shed some huge light on an area that we didn't fully understand. So, just fangirling a little bit, I would say I'd go back in time and meet Gregor Mendel, but I don't know that he would want to talk to me for reasons. All right, let's talk about alleles. So what did Mendel really do? Well, when he was looking at these different characteristics and breeding these pea plants, he was determining what we now call alleles. Alleles are alternate versions of a gene, okay? For the most part, Your traits are determined by the alleles that you inherited. I have blonde hair and blue eyes, which means that I have a gene for blonde hair and a gene for blue eyes. We know that these alleles occur on homologous chromosomes at particular spots that we call the gene locus or multiple gene loci. When your chromosomes are duplicated during the S phase of interphase, your sister chromatids have the same alleles because they're copied, they're identical to one another. And your homologous pairs can carry alternate versions of those genes. So someone who has brown eyes, my mom has brown eyes, actually, she's a great example of this. I have blue eyes. Brown is dominant to blue, and so she actually carries both copies of that allele. She has a brown allele and a blue allele, and that is how I was able to inherit that from her, okay? We understand this now, and these are the vocab we use now. Darwin called these factors and characteristics and things like that, okay? When we look then at these alleles, we can actually determine an individual's genotype and a phenotype, okay? Your genotype is going to be the copies of the alleles that you receive at fertilization, okay? We know that haploid cells, a zygote is formed when haploid cells are going through the process of fertilization. A sperm and an egg come together to create a new diploid organism. Again, this wasn't fully understood when Mendel had kind of came up with this whole idea here. But we know now that if you inherit 2 copies of the allele, there are a couple of different ways those alleles can be inherited. You could be considered homozygous dominant, meaning that you have a dominant gene for both alleles. You could be homozygous recessive, meaning you have 2 copies of the recessive allele. Or you can be heterozygous, where you inherit 2 different alleles, okay? We collectively are not going to see big A, little a, big A, little a, okay? We are going to say homozygous dominant, heterozygous, homozygous recessive, okay? Those are our allele combinations. That is what is telling us. And it doesn't necessarily matter if it's A, B, C, D, whatever, okay? Because if I say homozygous dominant, that means that you are going to inherit 2 of the same dominant alleles. If you're heterozygous, you're inheriting 1 of each, because hetero means other or, of each. And then homozygous recessive, you're going to have 2 copies of the recessive allele. Homo means same, zygous refers to the zygote, so that is what that is, okay? Your genotype are those alleles. Your genotype determines mostly your phenotype, okay? If I dyed my hair purple, that would be my physical expression of my hair. That is not my genotype being expressed, okay? I still have blonde hair, but it might not look the same. So your phenotype is the physical expression of the genotype. So if you are an individual who has the dominant allele, and it does not matter if you have 2 dominant alleles or 1 dominant allele, so if you're homozygous dominant or heterozygous, you are going to have the dominant trait. So in the example here, it's saying that if you are homozygous dominant or heterozygous, you have normal skin pigmentation. But if you are homozygous recessive, you have something called albinism, okay? That's that lack of pigmentation, and it results in skin that's very, very pale, white, hair that's white, eyes that can be very pale blue, and it can result in a lot of other things. People with albinism often have visual issues. Obviously, they're more prone to things like sunburn and that sort of thing, but it's because they happen to inherit 2 recessive alleles. So when we talk about allele combinations, this is some really big vocab here, and these don't go away. We talk about these all the rest of the semester. So don't lose genotype and phenotype. Genotype is the genes, phenotype is the physical, all right? So when we get into Mendel's actual experiment, right? How did he do? What did he do? How did he control this? Because it has to be a controlled experiment if it's following the scientific method. So the peas are fantastic for this because peas are self-fertilizing closed flowers. Now, most flowers that are open are going to have some sort of pollinator, a bee or a fly or a butterfly or something that comes along, picks up pollen and carries it to another flower, eats from that flower, drops the pollen off, and so on and so forth. Peas don't do that, okay? Peas self-fertilize and as such are considered true breeding if you just kind of let them do their thing. Their flowers are completely sealed and have to be cut open in order to then manipulate it. This means that Mendel was able to control who was making babies with who in his pea garden, because he was the one who had to physically open the flowers, and what he would do is he would remove the pollen producing parts called anthers, and then he would use that to pollinate the female part of another flower on a different plant, and see what happened when he planted their offspring, when those seeds were formed, okay? And so this was really huge. So if he had chosen any sort of flower that was open, it couldn't have been a very controlled experiment. You don't know if they're wind pollinated, if there's bees, if there's other bugs, whatever it might be, and it's really kind of hard to control that. But because these flowers were closed, he was able to control that. He also took incredibly careful, large records of his experiments. And he didn't just do these experiments once. He did these experiments thousands of times, cross-pollinating pea plants by hand, okay? Again, 1860s, right? Living in a monastery. It's not like he was out partying or going on TikTok and wasting time on the interwebs, right? That wasn't a thing. So this is what he did. This is what people did. So what did he find? Well, what he did is he would choose what he considered true breeding lineages. So when we go back to his traits that he was looking for, if he saw peas that were short, if he let them self-pollinate, they always produced short offspring. And then the tall plants would always produce tall offspring, and so on and so forth for each of these traits. So he decided to choose, and he did this for all of these. We're going to focus on seed color, I believe, or pod color. But he chose a trait and he started crossing the plants. He goes, look, if this plant is from a wrinkled variety and has always produced wrinkled seeds and this one's always produced round seeds, what happens if they get crossed? And that's what he did. So when he's doing what we call now monohybrid crosses or single hybrid crosses, he is taking the purebred parent generation, those true breeding individuals. So in this case, we're saying a green pod and a yellow pod. Green pods always produce green pods. Yellow pods always produce yellow pods. And what is he doing? Well, he's crossing these two flowers, okay? And he allows the first generation to grow. He gets the seeds from his cross. He plants the seeds in the generation. And that first hybrid is called the F1 generation. That's our first hybrid generation. What he did then is he didn't look and go, oh, well, green destroys yellow because all of the offspring are green from this cross. Okay, eat all of the green peas and be done. Because Mendel was a scientist. He didn't do that. So he actually took these first generation plants, which all turned out to be green, and he allowed them to self-fertilize, thereby crossing an F1 individual to an F1 individual. As a result, his second generation, so when he planted the seeds from that F1 cross, that second generation, the yellow pea pods showed up again. And what that told him is that green did not destroy yellow, but instead it masked it, which is where we get that idea of dominant and recessive alleles. So now we would say that a green pod is homozygous dominant if it's from the pure breeding lineage, and a yellow pod is homozygous recessive if it's from the pure breeding lineage. Their first generation crosses are all going to be heterozygous. And then the second generation, those F1s that crossed to produce F2 offspring, those F2 offspring do produce yellow individuals. So why does that make sense, right? Well, if we do something that's called a Punnett square, what we really see is that if I have an F1 gamete, that gamete can inherit either a dominant allele or a recessive allele, because that's what the parent has to give, right? It has a dominant trait and a recessive trait, because that individual is heterozygous. Okay, their gametes are going to be either dominant or recessive. And so we had these individuals that produced offspring when they were self-crossed that had either homozygous dominant, because in a Punnett square, let me draw. Hold on, I have to click a button over here. Let me draw. Pen. Okay. So I always do Punnett squares where I set up the female on the top and the male on the side. Okay, strong recommendation because you will be asked to do Punnett squares on the test. When you do that, you always put the dominant allele first and the recessive allele second, especially if you're doing like heterozygous crosses like we are here. And we would see that this dominant G and this dominant G results in an individual who is homozygous dominant for that trait, okay? If I say I'm crossing again, I'm going to go all the way across. My first individual here is heterozygous. We go all the way down and across. My second individual is heterozygous, and then across and down. My fourth individual is homozygous recessive. Now, it is not always going to be a 1 to 2 to 1 genotypic ratio, which is also a 3 to 1 phenotypic ratio. Absolutely not. If you cross 1,000 pea plants, sure. But if we do one cross, like I might get all green peas. I might think the yellow is gone. But again, Mendel did more than that. So how do we set up a Punnett square? Let's just do another example here before we get into the next thing so I can talk about these ratios as well. So if I cross an individual who is homozygous dominant for whatever trait and homozygous recessive, okay, if I say that this is the female and this individual is the male, so my dominant individual is my female, my recessive individual is the male, I would set up my Punnett square in such a way that I would have one dominant allele able to be donated, right? Because this individual can donate a dominant or dominant, and then recessive or recessive. And I know this is only 2, but think about it this way, right? This is going to get replicated, and we're going to end up with 4 large H's, right? Well, it doesn't matter what gamete I look at. They're all going to inherit a dominant H. So I'm a little less worried about that, okay? But if that helps you, do it. Now, again, we would do the cross, and I would get one individual who is going to inherit the dominant allele and then the recessive allele. A second individual, dominant and recessive, dominant, recessive, dominant, recessive. And we always write the dominant first because the dominant is going to mask the recessive when it's present. This tells me that I have a phenotypic ratio, okay, of 100%, let's say, red. Maybe red is dominant, okay? Maybe white is recessive. I have a genotypic ratio that is, I like to do my genotypic ratio like this. So where I have each of my possible genotypes written out. And so I have blank number of homozygous dominant, blank number of heterozygous, and blank number of homozygous recessive. I don't have any homozygous dominant. I don't have any homozygous recessive. And I have 4 heterozygous, okay? I can do the genotypic ratio. This did the phenotypic ratio for us. Physically see it. But remember, the phenotype doesn't always match the genotype if we are talking about if they have a dominant allele. My genotypic ratio for my green and yellow cross here would be 1 individual who's homozygous dominant, 2 individuals who are heterozygous, and 1 individual who's homozygous recessive. And so we just count, right? 1, 1, 2, 1. And again, this is not going to always be exactly this case. And why does that come into play? Well, when we do a 1-trait cross, this monohybrid cross, we don't always know if an individual is homozygous dominant or heterozygous, and they have the same phenotype, right? If you're a green pea pod, I don't know. I'm going to just eat the pea pod, right? If you are a yellow pea pod, I know that you're homozygous recessive for your genotype because your phenotype tells me that. But if you have the green phenotype, I don't know what your genotype is. So what happens? Well, you can do what's called a test cross, okay? So no one's going to get mad if you're crossing pea plants, okay? You can do a test cross with an individual to determine if they have 2 dominant traits or a dominant and a recessive trait. If I cross an individual that's green with a yellow pod who I know is homozygous recessive, okay, and all of my offspring are green pods, I know that my parent has to be homozygous dominant because all of my offspring are heterozygous. But if I cross an individual who's heterozygous with an individual who's homozygous recessive, I get a 50-50 split. Because if I do that Punnett square, right, my one parent is heterozygous, so capital G, that dominant allele and a recessive allele. Either one of those could be inherited by a gamete. And my homozygous recessive individual is going to donate a recessive allele to either of the gametes. And so I get one individual, my first individual chance is potentially to be heterozygous. The second is to be homozygous recessive. The third is to be heterozygous. And the fourth is to be homozygous recessive, giving me a 50-50 split of both my phenotypic and genotypic ratios. If you need more practice with Punnett squares and determining phenotypic and genotypic ratios, there are worksheets available on Canvas. I would suggest doing that, as well as watching the Amoeba Sisters Heredity playlist, specifically their one on monohybrid Punnett squares. Okay? What did this do, though? Doing these first crosses here allowed Mendel to determine his first law. Mendel's first law is the law of inheritance. The law of segregation. Law of inheritance was Mendel's things. The law of segregation explains meiosis before we knew what meiosis was. And meiosis just further supported Mendel's law of segregation. Because what ends up happening here is Mendel says, listen, each individual has two factors for each trait, which we call alleles now, okay? The factors segregate during the formation of the egg and the sperm. The alleles are split out into the individual gametes. The gametes get one factor from each pair of factors. Your gametes are haploid, whereas your individual cell, your parent cell, was diploid. And then fertilization recombines these haploid gametes into a diploid organism. Mendel came to that conclusion without understanding meiosis at all. Okay, meiosis was not understood. In fact, meiosis was developed much later, and it actually helped develop the chromosomal theory of inheritance as well, based on Mendel's laws that helped us understand meiosis. And meiosis just supported it. Isn't it just, I think it's just beautiful. I love that, like, this was figured out, one, before the communication technology we have today, two, before we understood a lot of the stuff that we understand now, and now we don't even teach it in that order because it's so out of the norm, okay? So, this is what Mendel determined by doing his crosses with his peas. But again, this man did not just stop here, okay? Mendel went even further. So he didn't just do monohybrid crosses. He actually also did something called dihybrid crosses. And dihybrid crosses is when Mendel looked at two traits and he said, okay, well, what happens if I cross a tall plant with green pods, which I know are both dominant, right? That's going to be a true breeding lineage. So those are going to be an individual that's homozygous dominant for both tall and green, okay? With a short plant with yellow pods, which we know is homozygous recessive for both of those traits. Now, your F1 plants are going to all be heterozygotes, because if I cross an individual, right, T with G, that's all that their eggs are going to get, okay? Let me do another line here with TG. I am going to end up, can I undo that? No, I have to erase it. Hold on a second. Eraser. Okay. I am going to end up with offspring that are heterozygous for tall and green, which we really understood even if we did the monohybrid cross, right? All of my F1 generation, all of my first hybrids are going to be heterozygous for this particular trait combination. But what happens when I let them self-fertilize? Now, there's two possible results for this, okay? I could end up with tall plants with green pods and short plants with yellow pods, and that would be what would happen if the dominant factors stuck together. Or, if the four factors segregate into the gametes independently, independent assortment, we would actually see four phenotypes present as a result. So what happened? Well, that's what happened. Mendel's second law of inheritance is the law of independent assortment, and it explains that if I have an individual who is heterozygous, I'm going to end up with a gamete that is going to carry the dominant trait for both characteristics, the dominant trait for one and the recessive for the other, the recessive trait and the dominant trait, and then 2 recessive alleles. And the reason that works is if I were to do what's called the FOIL method. So if this individual is heterozygous for round and heterozygous for yellow, which is using R and yellow then as our letters here, my first potential gamete would inherit the dominant trait for both. My second would inherit the recessive and the recessive. And actually, I did my FOIL method a little wrong. Hold on here. Let me make sure I make this very clear for you guys. I'm used to writing this on a board and not on the screen, and so doing it on the screen is a little bit funky. So I'm going to cross this big R to the big Y, and that's going to give me my first gamete here, right? I'm also going to cross the big R to the little y, and that's going to give me my second gamete here. Stop. Computer. 2, right? 2. Then I'm going to cross my little r to both of the y's. 3 and 4. And so I end up with the random segregation independently of these particular traits. And the reason that Mendel knew that this occurred and the reason that he came up with this law of independent assortment is because he saw these dihybrid crosses that support this second law, where he is going to see that of his potential offspring from his F1 crosses, if he did 1,000 plants, okay, it's going to basically boil down into 9 individuals who are tall and green, meaning they inherit at least one dominant allele for both the tall and the green trait. 3 individuals who are tall with the yellow pea pods, so they inherit the dominant for tall and the recessive, 2 recessive, homozygous recessive for yellow. 3 individuals who are short and green, and so they inherit the recessive alleles for height and then a dominant allele for color, and then 1 individual who is going to be recessive for both. Now this is all randomized. This is just going to be luck of the draw, and every fertilization event has an equal opportunity to result in these particular offspring, which is why when we see things like, I'm going to talk a little bit about some of the human factors here. But say something like cystic fibrosis. It's a recessive disorder that is carried in human genes. Individuals do not have a 1 in 4 chance to have a baby with cystic fibrosis, and if they have that baby with CF, then they don't have any more chances. Each time they have a baby, it is a 1 in 4 chance for CF. It's not like you had one, and so the next 3 babies that you have can't have CF. Every single time it's the same chance. And so when we talk about like this 9 to 3 to 3 to 1, again, it's when we talk about large batches, large sizes of things. We're not just looking at a single chance. If I were to plant 16 seeds, I guarantee you this would not be the outcome, because fertilization has the same potential outcome for every single fertilization event. Okay? I need you to check your understanding. I think this is where we're going to end today. Check your understanding, and then when you come back next time, we're going to talk about how Mendel's law applied to humans, and then how things don't fit within Mendel's law for the second half of our lecture on genetics. Have a great rest of your day, and let me know if you have any questions.